My name is Michelle Rathman-Josserand. I'm a research associate at L'Oréal, and I'm specialist in stem cell biology. I was hired by L'Oréal in the year 2000, and since I've worked in the area of skincare and also tissue engineering, and the focus of our work is really to understand the regeneration of the skin and the hair. This story began just with basic studies of comparisons of young skin with older skin. We have donors from 18 to 70 or 75 years old. We recover this skin and we can compare them. We can look at the proteins they express, we can look at the genes, and we can also look at their function. This is a test that we call a colony forming efficiency test. It's a test that allows us to quantify the regenerative cells that we fish out of a human skin sample. So we seed the cells at a very low density and we let them grow. And it's only the stem and the progenitor cells, which are their descendants, that are capable of dividing and multiplying thousands of times, a sufficient amount to form a colony that we then can color and we can see with our eye. And what we've seen is that with time, in fact, we're not losing the numbers of these regenerative cells. So that indicates that our stem and our progenitor cells are still there, but obviously they're not doing their job. Because what happens as we get older, well, our skin becomes thinner, we lose elasticity, also it heals less well. And if we take tissue samples and look at those, we can also see that the living layers are decreasing. So the regenerative function, that's clearly changing, but the cell numbers are the same. One thing that we've developed in our laboratory is reconstructed skin models that help us to model skin aging. We can actually fabricate, we can build skin. And in doing that, we can build a model that's very regenerative, like young skin, or we can do a model that has reduced numbers of these cells. We put in place a tool that allowed us to screen a certain number of molecules, and one of the active ingredients that came out of that, and it's cellules, they're dedifferentiated stem cells from rose. We showed that it can increase the expression of certain growth factors that are produced by the dermis and that are very important for the regenerative process of the epidermis. And then we tested it also to see the effects on our reconstructed skin sample. And again, we saw a beautiful reconstruction of our epidermis with lots of living layers, but the nice differentiation, so it looked physiologic. It's a very long process. And so that's what motivates me, and I think that's what motivates the team. Boost your energy for, another 5-10 years. So.